we have two main mechanisms that we can use to defend ourselves against pathogens: non-specific immunity and specific immunity. They have similarities and differences, right? Similarities are, of course, they're both helping to fight off pathogens in some way, shape, or form. Difference would be specific immunity is specific, right? This is going to be a defense that's directed against a very specific pathogen. With our non-specific defenses, we could use them to fight viruses or bacteria, and we didn't care whether the bacteria was Salmonella or E. coli. It wasn't even species specific, let alone, organism specific. But with specific immunity, we're going to look at how we can use our immune system to fight off one specific foreign invader. When it comes to our specific immunity, what works against influenza virus will not work against coronavirus. It's very specific. Another difference between nonspecific and specific immunity is that specific immunity has memory. When we are re-exposed to the same pathogen, the body recovers very quickly, and there are usually no symptoms associated with encountering that pathogen. This is through the use of memory cells. So we have certain lymphocytes, memory B cells and memory T cells, that basically it's their job to stick around for a long period of time. And if we encounter the pathogen again, they very quickly start up our specific immune response. And it's so quick and it's so effective that we don't even know that we were ever encountering the pathogen. Before we can talk about the specific immune response, we need to make sure we understand the terms antigen and antibody. Antigens are molecules on foreign material that cause you to make antibodies. So when we look at the surface of the virus in the picture here, we can see those orange and pink things on the surface of the virus, and those would be antigens. When our immune system cells bump into those antigens, it's going to cause us to make antibodies, and the antibodies are going to be used to attack the antigen. With our specific immune response, we are going to use our B cells and T cells, which are cells that can recognize and bind to specific antigens. And they are going to do this by using special receptors on their cell membrane that can recognize that specific antigen. Each and every individual T cell and B cell carries its own unique set of receptors for recognizing a specific antigen. So to kind of say this another way, the body's ability to recognize specific antigens on specific pathogens, then respond by making specific antibodies that only help to kill that one specific pathogen, is what makes the specific immune response specific. Before we can look at how the specific immune system kills off pathogens and gets them out of our body, we need to understand how T cells and B cells have the capabilities that they have. Every T cell and every B cell has to be taught how to do two things. Number one, how to recognize its one unique antigen. So every single T cell and every single B cell has a receptor on its surface that's very specific to one antigen. So we have to go through that process of, if you will, teaching the T cell and B cell to recognize its antigen. The second thing every T cell and B cell has to be taught is how to recognize self as self and leave it alone. So basically, I like to say that T cells and B cells go to college to learn this stuff. T cells are called T cells because they go to college in the thymus. B cells go to college in the bone marrow. So both T cells and B cells are born, if you will, in bone marrow. So they both are a type of leukocyte, and leukopoiesis occurs in our bone marrow. So they're both born in the bone marrow. B cells stay in the bone marrow, while T cells will migrate to the thymus. But once the T cells are in the thymus and once the B cells are in the bone marrow, they kind of go through the same processes. So as I mentioned before, T cells and B cells have to learn two things, but now I'm going to give you names for these processes. They're learning self-tolerance and they're learning specificity. Self-tolerance is what it sounds like. This is the process of the T cells and B cells recognizing self as self and tolerating it, leaving it alone. Think about what would happen if T cells and B cells recognized healthy self cells as pathogens. Yeah, that's problematic, right? Because then we would be starting a specific immune response against our own healthy cells. So that's not something we want. In the thymus and the bone marrow, T cells and B cells are tested for self-reactivity. So basically, T cells and B cells are shown a self-cell, right? And then the professors, if you will, say, what are you going to do to this self-cell? And if the T cell and B cell leave it alone, then they pass that test. If the T cell or B cell attacks the self-cell, then the T cell or B cell will be eliminated because we don't want to release those cells into our circulation because that's how autoimmune disorders occur. So autoimmune disorders are what happens when our immune system is attacking our own cells. So type 1 diabetes, psoriasis, rheumatoid arthritis, multiple sclerosis, all of these are examples of autoimmune disorders. And they occur when the T cells and B cells attack our own cells. The causes of autoimmune disorders are actually unknown. There are some very strong cases that show that for particular autoimmune disorders, genetics is involved. There are other really strong cases that show for other specific autoimmune disorders that a pathogen encountered early in life can cause that disorder. So there's the verdict is not out yet on causes. The other thing that T cells and B cells have to be educated on is specificity. T cells and B cells aren't really taught specificity. Instead, this is something that happens genetically. T cells and B cells all contain potential genes, potential genetic information for making receptors capable of binding millions of different antigens. So think about that for a second. What I just told you is that inside just one T cell, we have the genes for that T cell to be able to recognize millions of different antigens, right? Including antigens that don't exist and never will. I find that mind-blowing, absolutely mind-blowing. The information storage that we have in DNA is absolutely incredible. Okay, so what happens is early in development, the T cell and B cell takes that portion of DNA that's able to tell it to recognize those millions of different antigens. And it chops it up and recombines it into this new genetic material that then is used to make a protein. That protein is the receptor, the very specific receptor that will allow the T cell or B cell to bind to its one specific antigen. This process is called somatic recombination. It's called recombination because we're recombining genes into new ways that produce unique receptors that will bind to unique antigens. Somatic is just referring to the fact that this is not a reproductive cell, so it's not like eggs and sperm. Let's take a closer look at this whole idea of somatic recombination. Don't forget that DNA and its genes carry the code to make protein. Those unique receptors on the surface of T cells and B cells are made out of protein. Therefore, we have genes that tell us how to make those receptors. Somatic recombination is taking the genes for the receptors and chopping it up and recombining it in unique ways. At the bottom of the screen, we can see a long string of colored blocks. And let's say this represents the genes in a B cell that can be used to make the unique receptor. We're going to chop these up and recombine them. Let's take a look at what that looks like. So up at the top of the screen, we can see the starting genetic material. But then in the table on the left-hand side, we can see examples of new genetic combinations that were produced through the process of somatic recombination. On the right-hand side of the screen, you can see the new receptor that was created based on the new genetic recombinations. So we have 3 different gene combinations on the screen, and because each of those 3 gene combinations is unique, they each produce their own unique receptor. This is how T cells and B cells are able to produce unique receptors against any antigen we should encounter, and then even some that we will never encounter. Once the T cells and B cells have completed this process of somatic recombination, they now have their one unique receptor on their surface. Once that's done, and also don't forget, once they're tested for self-tolerance, they're then released into the body where they go to the bloodstream and our lymphatic system, and it's their job to look for that one antigen that they've been trained to find. So as I mentioned, a lot of this is in the lymphatic systems like the lymph nodes, but it could be in the spleen or the liver. That's where these lymphocytes tend to hang out. One thing I wanted to note about the B cell receptors is that B cell receptors are even more special. T cells and B cells both make receptors for a specific antigen. But the B cell receptors not only can recognize their specific antigen, the B cell receptors are also antibodies. They're actually a type of antibody. So we say B cell receptors are antibodies that are acting as receptors. So this antibody is embedded in the cell membrane instead of being released in the body fluids to fight the pathogen. And antibodies is the name of the game with the specific immune response. The whole point of the specific immune response, the whole goal, is to make antibodies, because that's how we get rid of infectious agents, is with antibodies. Once the specific immune response is initiated, we're going to make millions and millions and millions of antibodies. And this is because antibodies are made by activated B cells. Right? So once we activate the B cells, they, of course, have that antibody on their surface that recognizes the antigen. They just have to make that antibody in abundance and release it into the bloodstream, and that antibody will go and fight the pathogen that has that antigen on the surface. But in order to get our B cells activated, we need our T cells first. So T cells activate B cells to cause the B cells to clone and make the antibodies. So like I said, that means we have to get our T cells activated first. So we're going to take a look at all of this process in more detail.